Our protocol assesses the effects of myeloid-derived suppressor cells and the tumor-associated macrophages on T cell-induced tumor cell apoptosis, alongside investigation of the underlying mechanisms of potential therapeutic interventions. This protocol directly evaluates T cell-induced tumor cell apoptosis with high sensitivity and enables longitudinal analysis and imaging of cell-to-cell interactions.
The efficacy of checkpoint inhibitor is restricted by tumor-infiltrating myeloid cells in certain tumor types. This technique could lead to the identification of novel therapies for these tumors. This method could not only advance cancer immunology research, but could potentially also provide insights into the immunodeficiency and autoimmune diseases.
For the activation and expansion of spleen-isolated CD8 positive T cells, first aliquot 1 × 10⁵ CD8 positive T cells in 50 microliters of eDMEM into individual wells of a 96-well U-bottom plate. Next, add 1 × 10⁵ suppressor cells in 50 microliters of eDMEM or 50 microliters of eDMEM alone into each well of T cells. Then add 50 microliters of freshly prepared activation medium and 50 microliters of eDMEM with or without the test reagents of interest to the appropriate wells, and place the plate in a 37 degrees Celsius and 5% carbon dioxide incubator with 95% humidity for 4 days. To set up a pre-activated CD8 positive T cell target cell co-culture, first add 30 microliters.